Okay, today we're going to talk about some small RNAs that are very important and very interesting. These include the microRNAs, which are encoded by our genome and have important regulatory functions, and the small interfering RNAs, which also can regulate gene expression at the level of RNA. Okay, and it's not just about the process themselves, which is very interesting, but how we can go and exploit these for our own purposes. Okay, so we're going to talk about two classes of these teeny-weeny little RNAs, which are very small, between 21 and 22 nucleotides each, in contrast to something like an mRNA, which is several thousand nucleotides. And the ones we're going to talk about are the microRNAs, which are encoded by our genome and regulate translation and mRNA degradation of our own mRNAs. And there's also the SI, or short interfering RNAs, which generally originate from exogenous sources, but have the similar functions in regulating RNA degradation and translation. Okay, it's important to realize that the microRNAs originate from our own genome. They're there in our genome and transcribed by our own RNA polymerase, whereas the silencing RNAs generally come from invading viruses and exogenous sources. Okay, importantly, what they can be used for is to regulate or knock down particular mRNAs that we're interested in regulating, their own targets naturally, but also ones that we can exploit experimentally. Okay, so this all starts with the most famous Petunia in the world, the phenomenon of co-suppression, which later turned out to be RNAi, is started with Petunia experiments where people were trying to increase the intensity of purple color in petunias by giving them extra copies of an important gene in the pathway that produces that color. So they had a light purple flower, and they tried to modify it genetically by giving it more copies of that enzyme. And instead of getting more intense purple, they could get lighter and stranger and weird pattern ones instead. Okay, so the enzyme involved is called chalcone synthase, and it unexpectedly resulted in white petunias and ones with very strange patterns on them, which is hardly what you expected when you were trying to produce more of your purple color. So this phenomenon was called co-suppression. Some picture here. Okay, here's a whole bunch of transgenic plants with the chalcone synthase. And so you can see that some of them are nice and purple, but others have lots of white spots and splotches, and they're mottled. And how on earth can you explain that? Especially if you think they're starting off with a thing that's already purple. So how do you get something purple to turn white? So whatever was happening was it was turning off not only the transgene that was supposed to make it more purple, but also turning off the endogenous gene, or the endogenous RNA, because it should have gotten, if not more purple, certainly not white. And that was the phenomenon known as co-suppression, where the transgene somehow interferes with the expression of the normal gene that's already present in the organism. Okay, so you'd think you would get more copies, more purple, but no, instead. So it turns out that this is the tip of the iceberg in terms of recognizing a phenomenon that's especially important in plants, but also can be exploited in people. And that's what's called the RNAi, or RNA interference pathway. Okay, so it turns out that there was something the matter with their initial construct, so that artificially they were producing a double-stranded RNA. And it was the double-strandedness of it that was causing this strange phenomena of co-suppression, where the normal allele is silenced as well as the one coming from the transgene. So this is the silencing, or siRNA, or RNAi pathway, which leads to reduction in expression not only of the transgene, but of the wild-type gene as well. Okay, so in plants, this is an extremely important pathway. It is a response to invading viruses, which are very often double-stranded RNA. So the plant is trying to defend itself against the viruses. So the first part of the mechanism here is we're going to use a cytosolic enzyme called dicer. And what it does is it chops the invading double-stranded RNA into pieces of about 21 nucleotides in length. This chopping then leads to the formation of RISC complexes, which is RNA-induced silencing complexes that bind to the matching RNAs and lead to their destruction. Right, so that's diagrammed here. We have a double-stranded RNA. We have dicer, and that's a cytosolic enzyme, and it's going to chop the double-stranded RNA into small bits. After the small bits exist, then they form a complex with other proteins to form the RISC complex, one of which is a member of a protein family called argonaute, which has an endonuclease activity. So in the process of forming the RISC complex, one strand is removed and the other strand is retained. The retained strand is the guide strand. That's the one that's going to then hunt down the complementary RNA and lead to its destruction. The other strand is the passenger strand. So the RISC complex will hunt down the complementary RNA and then, because of its endonuclease activity, chop it where it's complementary, and that leads to the degradation and destruction of the RNA that came from the virus. So I think I probably said this. You have to have the argonaute family, you have the passenger strand, and you also have the guide strand. And the guide is what determines where it will bind, because that's the one that's complementary to the RNA that you're trying to destroy. So it's going to hunt down the complement. And in the case of a viral infection, of course, these would be the RNAs that are produced from the virus. And so that's going to truncate your viral infection if you're destroying the viral RNA. So the RNA is chopped, and that's degraded. Okay, this process has other parts to it that we're not going to deal with right now. Okay, the RNAi response is started by presence of double-stranded RNA in the cytosol, presence of double-stranded DNA in the cytosol, presence of double-stranded RNA in the nucleus, presence of double-stranded DNA in the nucleus. Okay, so the answer is A. It's the double-stranded RNA in the cytosol. The dicer enzyme is located in the nucleus or cytosol. Okay, so this picture diagrams not just the double-stranded RNA in the cytosol, but what happens in our nucleus to create these microRNAs, which act in a very similar fashion, but not identical. Okay, so the right hand of the diagram is the invading virus, and that's going to trigger the RNAi response. It's going to, the double-strand RNA is going to be chopped by dicer and incorporated into RISC complexes for the purpose of cleaving the target RNA. In our own genome, however, we encode these microRNAs, which can have similar functions relative to either RNA destruction or attenuation of RNA translation. Okay, so our microRNAs are encoded by our nuclear genome. They are created by RNA polymerase transcription within the genome to form a precursor, which is a primary microRNA. And they form these little stem loop structures with little arms, which are then processed by an enzyme called DRASA in the nucleus, which chops off sort of the parts of the ends. And if you have these tandemly associated ones, it chops them into individual ones. So the DRASA complex is in the nucleus. At some point, that becomes the pre-microRNA. It's exported from the nucleus into the cytosol, where dicer finishes the work of creating a double-stranded, sometimes, often anyway, imperfectly base-paired RNA, microRNA, which then becomes associated with argonaute proteins, again to form RISC complexes. So these are very similar, and they can have two fates. What they will do is bind to RNAs that are also encoded by our genome, generally speaking, where they will repress the translation of those RNAs. They also have the possibility of binding to RNA and leading to their cleavage. But the most important role of these microRNAs in us is this 3' untranslated region binding there and reducing translation of the RNA that's complementary or almost complementary to the microRNA. Okay, so these are very often regulatory, and they are part of the same pathway in the sense that both use dicer, both will incorporate themselves into risk complexes, and they will form risk complexes associated with base paired to mRNAs, which then leads to their translational repression or sometimes to their destruction. And this is the endogenous pathway because the microRNAs originate from our own genome, not from some exogenous source like a virus. Okay, so here's another version of microRNA synthesis and processing, where your primary microRNA transcript is created by transcription. DRASA processes it initially in the nucleus. It's then exported, where it's subject to dicer cleavage, and then is loaded into RISC and targets a complementary mRNA. Okay. Yet another version of the same thing. Your primary mRNA, your pre-microRNA, dicer, duplex, RISC. Okay, so here's just another representation of the same process, which emphasizes the commonality of the use of dicer and the use of RISC in the cytosol between these two pathways, and the two different outcomes that you can have as a result. So the natural role is to regulate translation, and they do serve important functions in that regard. They're important regulatory molecules. They bind to complementary RNAs, usually in 3' untranslated regions. So the silencing and microRNA pathways both use RISC, DRASA, dicer, A and B, A and C. Okay, they both use RISC and dicer. DRASA is restricted to the microRNA pathway. MicroRNAs are produced from invading viruses, naturally occurring short hairpin RNAs. B, naturally occurring short hairpin RNAs. Okay, so scientists being as we are, what you want to do is use this to exploit for your own purpose. So it was immediately obvious that this could be used as a form of gene knockdown to reduce expression of something that you want to get rid of. So there's an RNA that you don't like. It doesn't have to be a virus. It could be something that's associated with cancer progression, could be something to do with genetic diseases or whatever, that you could basically make a fake RNAi that will knock down something that you want to either get rid of for a therapeutic purpose or to understand the effects of the absence of that particular function, whatever it may be. So these RNAi type experiments, knockdown experiments, especially in cell lines, are used extremely widely for those kinds of purposes. Okay, so the very best early experiments on this to prove that it worked easily was done with C. elegans, the worm. Okay, and this is a fabulous organism in general, but the experiments to show how RNAi worked were genuinely astonishing. Okay, so in terms of how you cause knockdown, how you can manipulate the system experimentally, you know that you can get double-stranded RNA into cells by transfection, no problem that way. The other way to make a double-stranded RNA that looks like a microRNA is to make a plasmid that will produce a hairpin-type RNA, and that will be processed by DRASA, etc., to produce the RNAi response. In worms, you can actually just inject it into the worm, and it works. The most sort of unbelievable version of this is that you can actually feed the worms E. coli cells that are producing the double-stranded RNA, and the worms will have an RNAi response against the complementary RNA, even after eating the E. coli. Weird. So in terms of what gets put into the worms or cells or people, you can use double-stranded RNA, plasmid that will produce a double-stranded RNA, and the idea is that your intent is to downregulate the target RNA either by destroying it or repressing its translation. Okay, so in terms of how to do this functionally, there's different ways. Plasmids are used to create short hairpin RNAs, but they're a little bit different from what we're usually used to seeing. So your idea is, I'll show the picture, I guess. Right, so here's what one looks like. Here's a plasmid that's sold for this purpose. You're going to basically take a short piece of DNA and clone it between the BAM and EXPRESS sites that are shown. It has an origin of replication. It has a candine for selection in E. coli. And what you're going to do is take a very short piece of DNA that's predicted to form a double-stranded hairpin RNA by cloning it into the sites. Now, the promoter that's used is actually usually a U6 promoter for reasons that really aren't important right now. Okay, so this is, for example, part of GFP that we're going to use to knock down GFP. And important is when you transcribe this, you make an RNA single-stranded that is self-complementary. That if you work out the nucleotides here, when you produce this particular single-stranded RNA, it will, of course, be equal to the upper strand, which is a non-template strand, and it will fold to form a hairpin, which can then be used to knock something down, in this case, GFP. Okay, so the idea is we're going to trigger the RNAi response by providing this RNA, this hairpin RNA that we've designed for the purpose. So what's going to happen to this once it's made? It's going to be processed in the nucleus by DRASA, also called the microprocessor complex. It's going to trim off the ends, chop off the loop, and then it's going to be in the cytosol for loading into RISC. So it's then presumably going to then hunt down its complement, in this case GFP, and knock it down. So the first sort of type of thing you want to do is to do this just to make sure it's actually going to work, of course. Okay, so this is what it looks like with this sort of experimental situation here. We have a cell line which has got GFP in it being encoded by a plasmid, and that's PEGFP is being produced. So GFP is produced in the cells, and you can see that they're glowing nicely. But then at the same time, you take those cells and add, in addition, a GFP encoding a short RNA that is expected to bind to, hybridize to, the eGFP RNA and silence it. So you can certainly see that these are not glowing the way the original ones were, saying that the silencing RNA that you designed and put in there is functioning correctly as desired to prevent production of the GFP protein in the manipulated cells. So this definitely works. Right, so remember we can see GFP by putting it in the light microscope, in the UV microscope, and it glows green. Okay. Right, so here's another example where the RNA was injected into worms. It wasn't transfected into cell lines. These are actual worms. That we've got the vector control, and it's expressing GFP nicely. And then if you put in the GFP silencing RNAi, the GFP signal goes away. This was done in spaceflight. I don't even know where I got this slide, but in the space flight, the GFP is being expressed normally and responds normally to its GFP RNAi and is silenced in that situation. So this works. All right, so GFP is nice, but what you want to do is have it work on cellular RNAs or whatever that you're trying to get rid of. So a more customary way to assess whether your RNAi experiment is working correctly is to use a Western blot to detect reduction in the target protein. And this actually makes more sense than looking directly at the RNA, because sometimes you don't have reduction in the RNA, it's just translationally repressed. But we certainly expect reduction in the protein, whether it's a translational repression or actual degradation. So if we look for the protein, and then we'll infer that the mRNA was destroyed or repressed. Okay, so this kind of thing needs controls, and this shows kind of what goes on here. Okay, the first lane is a negative control where you have cells which have no silencing RNA and no eGFP plasmid. So all the cells that are being shown have the plasmid that expresses GFP in them. So we're going to be looking for GFP expression or reduction depending on the situation. So of course the cells don't have GFP in them naturally, so if you probe with a GFP antibody, you will not see any lane. Okay, the next two lanes are where you have the GFP plasmid is present, but there's also a silencing RNA been put in there, but it's one that does not bind to the GFP RNA. So this is kind of an irrelevant other gene, which has been, well, the intent is to, as a control, to show that the silencing RNA in and of itself doesn't hurt anything, that it has to be specific. So this hybridizes to something else completely. And of course has no effect on the amount of GFP protein produced, because you've got lots of GFP. Okay, so the next two lanes are where you've got cells with the eGFP expressing plasmid. So they're supposed to be able, they've got the RNA there, and but they're also treated with an RNAi that's targeted to the GFP RNA. So this one is expected to bind to the GFP RNA and reduce its expression, either repressing its translation or destroying it. And you can certainly see, if you look at the bands, that yes, there's much less GFP being produced when you have your GFP plus the siRNA that's targeting it. So yes, this works. Right, so with all these controls in place, with our negative controls and our RNAi directed against something else, and then the one that's expected to reduce GFP, we can see that yes, this silencing RNA did reduce GFP expression. Okay, now there's some controls that have to be done here, and so this is more of introduction to some experiments we'll be seeing mostly next time. Okay, so the idea is, like, do you really believe what you see? The idea is you've got these cells, you mash them up, and you make some protein extracts and run them on a gel. You try, but you can't know that you're loading the same amount of protein on the gel. What if there's no protein at all on the gel? That sample is just messed up. So a darker band might just mean that you put more cell extract in one lane than another. A lighter band might mean that you messed it up somehow. So how do you actually make sure that when you're comparing band intensities on the gel, you're comparing things that actually are meaningfully different from each other? And that's where the sort of normalization kind of comes in. Okay, so what's typically done is to include a loading control on your blot or a parallel blot. So what you're going to do is you want to compare the protein of interest, which you're trying to see changes in, to another one that's not expected to change. Okay, so that's what's called a loading control. Things that are commonly used for this are actin, often used, and other things, metabolic enzymes like glyceraldehyde 3-phosphate dehydrogenase. Those are considered housekeeping genes in the sense that they're expressed fairly equally in most cell types under most conditions. So if you're looking for changes in something else, doing it by comparison to these things that are not expected to change, you can believe more that what you're seeing as a difference is real, rather than just some artifact of your messy old experiment. Right, so here's an example. Ignore the figure on the left, that's RNA. Okay, what this is, the SI control, which is to say there's nothing targeting anything. It's just a control of irrelevant scrambled sequence or something. Okay, so what you're going to be looking at, you're going to try to compare the control here, which has no silencer in it, to something that is supposed to silence something. Okay, so you can see in the Western on the left, we're going to look at protein RRM2 and also actin by comparison. So you can see if you look at RRM2 and that you've got actin in there, that there's more RRM2 than there is actin. Right, so that's fine. You can calculate some ratio based on the band intensity of those two things. But the important one is the next one on the right, where you've got a silencer that's directed at something, and what it's going to try to be doing is cut down the RRM2. And you can see that the band is less intense in the silenced version, but how would you know that that's real versus some loading artifact of some sort? Well, the actin tells you that it's not an artifact, because the actin looks pretty similar between the control and the one with the silencer, versus the RRM2 is obviously looking at it much more in the control versus the silencer. So this will give you some comfort to tell you that yes, it really is working, that there's nothing wrong with the extract, that there actually is a reduction in this RRM2 protein as a result of the presence of this silencer. So it's a very important control to have something to compare to. think this is what I said. Now, the way people actually deal with these numbers varies. Sometimes they'll actually say what the actual ratio is between the intensity of the actin or whatever the loading control is to the protein of interest. But sometimes they don't. Sometimes they just say whatever that ratio is, and they're going to call that 1, and then look at deviations from 1 in other experiments. So you have to, different people do it different ways, but this normalizing, just to say whatever the ratio is, that's 1. And we're going to look at deviations from that in the treated one, is what that's about. We'll see some examples. Okay, the other thing we have to be able to deal with is quantitation of RNA, whether the RNA has disappeared or is simply not being translated. And that means that we're going to have to be able to do some PCR using RNA. Okay, so qPCR is quantitative PCR. One version of this is RT-PCR, which starts with an RNA template. Okay, so starting with this, PCR is not quantitative usually anyway, but you still want to use PCR. So what quantitative PCR is basically monitoring the reaction as it goes along, and you'll be able to see that from what I'm going to show you in a second. But what's important is it's not quantitative because the amount of final product does not depend on the amount of template that's present. So it's not a quantitative procedure without some variation. Okay, so the RT-PCR part, that's actually a fairly simple part of all this. You get an RNA template and easily convert it to a cDNA using reverse transcriptase. So remember that reverse transcriptase is an RNA-dependent DNA polymerase, and if you give it a primer, it will copy your RNA to form a, single-stranded cDNA, but that's fine. Once you've got a cDNA, then it's DNA and can be amplified by PCR. And then we have to worry about the quantitative part. Okay, again, the quantitative part is going to involve measuring the amount of product during the reaction after each PCR cycle. All right, so the idea is you're going to follow it as it goes along, and then you're going to compare the different samples to each other in terms of how long it takes them to get to a certain level. All right, so I think this slide exemplifies it fairly well. This is a qPCR reaction, and the different curves represent the same template which you've put in at different concentrations. So it starts with 1 microgram as the most, going all the way down to 0.1 picograms as the least amount of template of the same thing. So if you look at these reactions over time, if you look at the very end, after 35 cycles, you get basically the same amount of PCR product, irrespective of the amount of template. The 0.1 picogram gives almost as much product as the 1 microgram does. So it's clearly not quantitative. It's precise, but it's not quantitative. Okay, so what makes it quantitative is that these curves are being acquired or observed at every cycle of the reaction. So what's happening is the instrument is basically quantifying the amount of fluorescence in your probe at each cycle in the reaction. So in this you can see the 1 microgram, it starts shooting up very early after only a few cycles. And of course the others come shooting up later according to how much template there was in the first place. So you can see clearly in this, we can distinguish those with high concentration of the template from those with low concentration of the template, according to when in the whole process you get discernible amounts of product. So without being quantitative about this, what's set is an arbitrary threshold, or a reasonable threshold, which occurs during the exponential phase of the reaction. And so the samples are compared according to how many cycles it takes to achieve that threshold. And so even though the final amount of product is basically the same in all cases, the number of cycles it takes to get to that threshold will vary quantitatively with the original quantitative of template. So there's a lot of calculation that goes into this, but that's how qPCR works. And it works with a double-stranded or DNA template, and also works with a DNA template that's been created by reverse transcription of RNA. Right, so here's another picture where this set of samples, obviously, the two sets of samples are very different in terms of their initial concentration, whereas the product amount at the end is basically the same. All right, so in the experiments to follow, we will compare the behavior of our gene of interest compared to normalizers for RNA and protein, which are not expected to change. And so what you end up looking at is bar graphs, which actually reflect quite a lot of work. It's this very simple bar graph that comes from this enormously complicated PCR reaction. Okay, examples and worksheets next time. Okay, so we will deal with this next time. I will send this out to everybody.